Hi, everybody. Welcome to another Chats with Chaudhry. I hope you're safe and well. Today, I'm delighted to be joined by two scientists, not one, from the Waters Corporation, and we are going to be talking about glycan analysis and peptide mapping. And I'm delighted to be joined by Xiaoxiao Lo and Wenjing Li. And I hope I've got the names correct. So, Xiaoxiao, Wenjing, how are you both?
Good.
Yeah, very fine. Thank you.
Good. I'm glad to hear it. And where are you guys calling in from today?
I'm calling from my office in my apartment
And where is that? In Boston?
in Natick.
All right. Okay, cool. And what about you, Wenjik?
I'm actually in Water's office right now, one of those conference rooms. So I got some lab work to do today, so I'm popping in.
All right, fantastic. Well, I hope it's sunny and the cold weather has gone now in Boston. Because I know you had snow certainly last week when I interviewed someone near Boston. So hopefully the snow is all melting away. It's no snow in the UK, it's just wet and rainy this evening here in the UK. But anyway, so today we're going to be talking about glycan analysis and peptide mapping. But before we start talking about that, could you guys give me a quick overview of the areas that you're responsible for and also a bit about your backgrounds as well? Who'd like to start? Xiaoxiao, would you like to start?
Sure. So we're senior scientists in chemistry R&D at Waters Corporation. So we're in charge of doing the research and development for some consumables that maybe improve the technology for sample preparation and analysis. Yeah, so some of the background of myself. So I got my bachelor degree at Fudan University in China in chemistry, and then I come to US, Michigan State University to do a PhD. And all my PhD work is focused on the plant metabolomics. And then I move on to Waters to join this group to do a more interesting research for the protein analysis.
Fantastic. And what about you, Wenxing?
Yeah, so I joined Waters about 2 years ago. So I work in the same big group as Xiaoxiao. We basically help design, develop, and also evaluate a lot of chemistry consumables, most often for LCMS analysis, but also many other, downstream analysis. We help develop, columns, reagents, vials, and I recently focused on the peptide mapping workflow, which is a sample preparation technology for mostly biopharm customers. So we help to simplify and streamline the workflow so they can do the LCMS analysis much easier.
Fantastic. All right, so let's talk about glycan analysis and peptide mapping then. So first question, really basic one. So what is peptide? What is glycan analysis and why is it important? And what are the main challenges that researchers face when using it?
Sure. So for glycan analysis, we all know that after the protein has been expressed from the cells, sometimes the cells, the host cell itself will put some modification on the protein to try to either improve its stability or to do some to help it do some immune response for it. So the glycosylation is one type of this post-translational modifications that take place on this surface of this biotherapeutics. So usually because it's in charge of the protein stability, immune responses, so we treat it as the critical quality attributes. So that's why scientists need to monitoring those kind of modification during different stage of the drug development. So that's why we are interested in this glycan analysis. Why it's important, as I said, is the quality attributes. So we need to monitor it. So we need to find some tool to do the accurate analysis of this stuff.
Right.
And
Sorry, I stopped you. So what were you going to say? Sorry.
I just see then we move on to the challenge part. So one of the big challenge for the glycan analysis, especially the released glycan analysis, is the sample preparation. So imagine that we had the protein and had the modification on it. To analyze this modification, first we need to find a way to just cut off the modification from the protein itself.
Right.
And then we need to purify those this glycans from the sample matrix and then put some tag on it to help us do this analysis, and then move on to the instrument analysis part. So this whole process usually take a long time. And also during this long time sample preparation, you may lose some of your sample during this analysis procedure. So this sometimes a big challenge for our glycan analysis.
Right. And how do researchers then overcome those particular challenges?
For our company, we developed a full workflow and glycan analysis kit to facilitate those glycan analysis. So it can ensure us to do those whole workflow within 1 hour and to get a more reproducible and accurate result after doing this sample preparation.
Right. Okay. And let's talk now about peptide mapping. So why is that important, and what are the challenges researchers face doing that?
Right. So peptide mapping, by its name, you can tell, is to generate a map of peptides which correspond to the protein that you're studying. Usually, this is through a enzyme digestion step. So most often people use trypsin because it gives a very reproducible and accurate digestion results. With all these, you can consider the mixture of digested protein, the peptides, as a fingerprint. So with this fingerprint, you will be able to tell the identity of the protein. So for biotherapeutic, you will be able to confirm the protein ID through, the map. And also, very importantly, many amino acids has some modifications on it. So one of the attribute that Xiaoxiao mentioned is glycosylation. So this is a post-translational modification that should be monitored carefully. And we also want to know which amino acid carries that modification. So peptide mapping gives the customer a chance to monitor where they are and how much is modified. There are many other, concerns with storage and stress conditions. So they would monitor the oxidation, deamidation through peptide mapping as well. So that's why I think people realize how important it is, and I think it's being done currently in the released assay for biotherapeutics. So you need to validate and prove that your therapeutic is correctly made and also carries high quality. So I think the challenge really is because you have to do such a careful characterization, you need to make sure your assay is reproducible.
Right.
Also, every protein is different, so you may have to optimize your sample preparation every time. So it's kind of like a lengthy, tedious step. What people usually do nowadays is try to adopt some level of automation,
right,
for method development or for large sample size. So it's not so rare now to see big pharma companies to use liquid handling to do all these sample preparation.
Right. Okay. And so you're saying the main way to overcome the challenge is through automation is the main way to overcome those challenges then?
So automation is one of the area that people have started looking because one, it could improve the throughput because you are conducting, hundreds of samples a time. And the other thing is because it's automation, you can avoid some human errors. So you don't have to worry about, padding procedures and transfer your method to different locations. So that's, a very significant benefit of doing automation.
Right. Okay. So in terms of these analytical tools, how helpful are they in terms of COVID research and vaccine development? Who would like to answer that?
Yeah, maybe let's start with the glycan analysis. So in the COVID research, one of the hot topic is the characterization of the spike protein.
Right.
So the spike protein usually has been covered by a lot of glycans. So for the spike protein itself, it's used as glycans as a shell to protect itself from the immune system for the host cells, right? And we also know that the spike protein will in charge of the like the interaction between the virus to the host cells. So because this protein has been highly glycosylated, we need to find a tool to help us understand and characterize those glycan shells on the surface of the spike protein. So that's why the glycan analysis is important and usually will be helpful to help us understand the spike protein. And for the vaccine development as well, sometimes the spike protein and the glycans is target for the vaccine development. That's why we also need to monitoring those glycosylation during the vaccine development for the spike protein and coronavirus.
Brilliant. And Wenjing, what about peptide analysis? Can that help in any way?
Right. So, peptide mapping could be translated into many different terms based on what exactly they're doing. So I've seen many papers published last year using the same technology as peptide mapping, but they call it glycoside mapping, which is, to monitor the glycosylation. So for COVID research, I think it's very important for people to understand where are these glycosides on the spike protein,
Sure.
and whether they have some biological functions as to how the spike protein interact with the receptor, how they can shield some of their epitopes to avoid neutralizing antibodies. So these could have a very fundamental understanding of the glycobiology. And on the other hand, we all know that mRNA vaccines are so popular these days, but we also monitor some other protein-based vaccines. And those vaccines are making, a highly similar spike protein as the SARS-CoV-2.
Yep.
And with those vaccines, so Novavax is one of the front runners here in US. Their protein-based vaccine is a mimic, and they have carried out very extensive, glycoside mapping on their vaccine candidate. And so I think this could be a platform to monitor, the quality of the vaccine, especially it's protein-based.
Right. Have you got anything else to add to that, Xiaoxia?
I think I have no more to answer. It's just like the released glycan, we can know what the glycan itself it is. And then for the peptide mapping to help to further understand the glycosylation of this spike protein, because there we can know that which glycan is on which side that modified on the spike protein.
Right. And what other areas can we see these tools being used in the future? What do you think the future is for these two sort of applications?
For the glycan analysis, it can be applied to a broad range of the biotherapeutics and the vaccine development. And also for the technique itself, as Wenjing mentioned, that automation is always a very hot topic in all of those sample preparation area. That, as I said before, for the glycan analysis, you also need a long time for you to do the sample preparation if you do it in lab by scientists itself. So automation is always a good way to help improve the throughput and to help people to get the result in a very fast, in a very short amount of time.
Fantastic. Yeah. Wenjing, anything else to add to that?
Yeah, I think with peptide mapping, it's always a popular, technique that biopharma uses. What it can play more maybe is to provide more information on the glycobiology side of things. I think with SARS-CoV-2, people started to investigate more glycoproteins, viral glycoproteins, things like that. And also what is very interesting to see is whether peptide mapping could be used for cell and gene therapy. I've seen some analysis on, capsid proteins of viruses. So those kind of assays could also help with, characterization, release, things associated with cell and gene therapy.
Brilliant. So I know you guys have done some papers on this as well, haven't you? So where can people get more information on both glycan analysis and peptide mapping?
For those kind of information, I think if you go to our Waters website, www.waters.com, we should have a COVID research hub for all the applications that we have done for the SARS-CoV-2 analysis, and as well as other applications that for either the glycan analysis or the peptide mapping analysis.
Fantastic. All right. Well, ladies, what I want to say is thank you very much for enlightening this non-scientist about these two topics. It's been really interesting. Thank you very much for sharing your knowledge. It's been lovely to meet you both. I hope, as I said, the winter snap has gone in Boston and you get lots of lovely weather and you have a fantastic, happy, healthy and successful 2021. So thank you very much for joining me today.
Thanks for having
Thank
us.
you.
Thank you. And thank you for watching. As Wenjing and Xiaoxia said, there is going to be information on the Waters website. You can check the link above the video. You can also, because they're both on LinkedIn, I know that because I'm connected with them, you can send them a message on LinkedIn as well if you've got any questions, or put some comments below the video as well, and I'm sure they'll be happy to answer them. So all that's going to say, as always, is thank you very much, Xiaoxia and Wenjing. It's lovely to see you, and thank you for watching, as always, and until next time, stay well and stay safe. Bye-bye.